Next up, we have Claire Grosset, who is a veterinarian who graduated from France as a diplomate of the American College of Zoological Medicine and is currently an assistant professor at the Université de Montréal and the Aquarium de Quebec in Canada. So, Claire, I believe we have your video to play.
Yeah,
Yes, you're correct.
Play the video. Okay.
Thank you. Hi everyone, thank you for the opportunity to present about the establishments of hematology reference intervals in big-bellied seahorses. I would like to start by acknowledging my co-authors, especially Dr. Jean-Philippe Frigon, who was the main author on this study and who is an intern in our service. So big-bellied seahorses are a popular species in aquaria. They are often affected by mycobacteriosis, and this disease causes a high fatality rate. Currently, there is no antemortem screening test, and so we wanted to determine whether heterophilic and monocytic leukocytosis could be present in those seahorses that are affected by this disease. So the first step before determining this parameter is to establish reference intervals for hematology in that species. Currently, there is no reference in Signateds based on the ASVCP guidelines, which are the guidelines recommended in veterinary medicine for hematology references. Hematology is a very easily performed test. It's minimally invasive and easily accessible to many clinics. Our objectives were to determine hematology reference intervals, and a secondary objective was to determine whether the Nat and Herrick technique would be similar to the manual counts, and we'll define that technique later. Finally, a secondary objective was to determine whether there would be differences associated with sex in seahorses. For our study, we had 38 seahorses housed at the Aquarium de Quebec. They weighed 6 to 16 grams and were 6 to 8 months old. They were kept in 4 tanks with nitrites, ammonia, pH, and temperature within the normal range for the species. They were all considered healthy based on the follow-up of their appetite, body condition score, physical examination, skin scrapes, and also necropsies performed in 5 individuals prior to the study. No mycobacteriosis was detected on necropsy at the beginning of the study. So first, we performed venipuncture from the ventral coccygeal vein, and then we placed that blood, which corresponded to 0.05 to 0.1 milliliter, into heparinized insulin syringes, and then into Eppendorfs with heparin. Here you see there was a range in heparin concentration because initially we had some problems with clotting. So we tried different concentration, but this didn't have any impact on the clotting problems. Finally, we decided to store the blood in warm conditions in basically our pockets, and that helped and resolved the problem of clotting that we had initially. So we collected 33 samples that were dedicated for the Nat and Herrick technique for 10 of them. The remaining 5 samples basically were clotted, so that's why we didn't use them. And for the Nat and Herrick technique, it's basically a technique that's used for exotic pets. We use a hemocytometer, and then there is a staining that affects the red blood cells and white blood cells. And this technique allows to count both the red blood cells and white blood cells under the microscope. This is a technique that's commonly used for nucleated red blood cells, like for fish, for example. We determine the hematocrit for each individual, the platelet counts. We determine the white blood cell estimate on a smear, and also graded the cell morphology. Regarding the statistics, we used the software RefAdvisor, excluded the outlier using the Tukey technique, and then we used a parametric method or box-plot transformation whenever pertinent to obtain reference intervals. Then we compared males to females. We used GraphPad Prism and used first a Shapiro-Wilk test to determine normality, And then because the data was not normal, we use a man-Whitney test. And finally, we compared the NAT and NERICS techniques versus the white blood cell count estimate on smears using a Spearman coefficient. The p-value was 0.05. Regarding our results, first, the seahorses tolerated the blood samples quite well. The only problem we encountered was some color changes in some individuals. At the site where the tail was basically punctured with the needle, some of the individuals had a discoloration of the tail distally to the venipuncture site. This resolved over time for most individuals. All seahorses kept a normal appetite, and none of them died during the study. There was no significant difference detected between male and females. Regarding our results, we conducted some special stains to better evaluate the cells present. What we found, first, regarding the erythrocytes, is that none of the stains actually stained the erythrocytes. For the thrombocytes, now you can see on the picture that the PAS stained with some purple dots, the cytoplasm of the thrombocytes. Now for the neutrophils, we had some staining with ALP stain. Also with PAS, you can see the purple cells, which means that they are stained positively. And we didn't have any staining with the toluidine blue stain. Regarding the lymphocytes, we had some partial staining with PES against some purple dots around the cytoplasm of the lymphocytes. And we had some positive staining with toluidine blues. You see these cells are quite blue. You can see them basically over here. That's a small cell. It's a lymphocyte here. And then the monocytes, which are here, were also stained by the toluidine blue. Here you can see the staining with the different special stains for monocytes. Now, regarding ALP staining, what we observed is there was moderate to marked staining in 90% of the cases for the neutrophils. So here you see a mark staining, which is very black, and here this is just a moderate staining. So just to show you that there was some variability depending on the smears. Now, regarding the cell morphology, we had slight anisocytosis for the red blood cells, no poikilocytosis, and polychromatophilia was present in 94% of the cases. We had no reactive lymphocytes and no toxic neutrophils. Regarding the Spearman coefficient, it was only 0.21 between the two techniques. And so that shows us that basically there was no correlation between the NAT and NERICS techniques and the white blood cell count estimates. So we would not recommend using the NAT and NERICS techniques for that species. Now, regarding our reference interval, here you have them detailed in the table. But what was interesting is we had a high range, high variability for the hematocrit. You see 27 to 67%, this is highly variable. Same thing for the leukocytes. One parameter that was really interesting is the neutrophil to lymphocyte ratios. It was basically between 0 and 1, which would be easy to remember. So basically we had either more lymphocytes or a similar number of neutrophils and lymphocytes, depending on the individuals. So why did we have so many variables? Well, the first parameter that comes to mind would be stress. These seahorses can be quite stressed during the procedure. You see these were performed without anesthesia to not have any hemolysis of the cells. And so that could have been stressful for them to just be restrained. Then we had some variability in the cell morphology, for example, the red blood cells, and this has been reported to be dependent on some water parameters or stress variables. We also encountered some difficulty with sample storing. We noticed that there was coagulation initially in the Eppendorf tubes when we wanted to perform the NAT and NERICS techniques. And this was basically resolved when keeping the samples in warm conditions. So it could be similar to the coagulation that we can see in people that have clotting problems and are sensitive to cold. Now, regarding the intervals that we established, we saw that there was a high variability depending on the individuals. Lymphocytes were the predominant white blood cells. And we can compare the hematocrit that we found to those previously showed in a previous study in erectus seahorses. They had a lower variability, but they also had a lower number of individuals and didn't use the ASVCP guidelines, so that might account for some of the variability. Probably there was a handling bias for some smears. Some of them were thicker than others, even though the same person did all the smears. But we could still rely on the neutrophil to lymphocyte ratios when using different smears. These would not be affected by the thickness of the smear. Regarding the staining, it was sometimes difficult to differentiate lymphocytes versus thrombocytes. And this problem could be resolved using a PAS with diastiles, which is a different staining, which could be used in this case. We also noticed some inclusions. I'm just going to move my window here. So here you see the inclusions in the cells. These were rectangular in shape, very variable in their localization in the cell. And so we think those were actually artifacts and not viral inclusions. Finally, we found a low correlation between the natriurex and estimate techniques. This could be due to some staining issues. For example, when we were reading the slides, we noticed that over time, some of the cells were losing their color with that staining. And again, this technique has not been designed to be used for fish. So maybe that's something that would need to be improved in some species of fish. Also, we noticed that some of the cells were really ballooning with that technique, and that might be due to different osmolarity of the blood of seahorses compared to mammals. So these cells were really enlarged, probably because of the osmolarity of the solution. Again, some other studies have shown that the validity of the natriurex techniques depends on the species, and so that might not be applicable to seahorses. Regarding the male to female comparison, we have the low population, so it would be needed to have a larger population to assess some more subtle differences. And those individuals were not sexually mature, so it would be interesting to repeat that study in older individuals. Finally, we hope that these reference intervals can be used for seahorses under human care, both in zoological institutions, in laboratories, and also in piscicultures. The perspective we have for this study is to compare subadults to adults and also healthy to seahorses affected by mycobacteriosis to see if we see a difference that would be clinically relevant. For this study, I would like to thank especially the clinical pathology lab of the University of Montreal, the Centre québécois sur la santé des animaux sauvages in Montreal, as well as the Aquarium du Québec, especially Isabelle Godet, who restrained the seahorses during the study, and you can see her on the video. Thank you so much, and I'm happy to entertain any question with my team.
Thank you very much for that wonderful talk. Does anyone have any questions? If you have a question, feel free to unmute yourself and go ahead and ask. While we're waiting for anyone else to ask a question, I'll kick it off. Claire, I was wondering how many different species tend to be impacted by this type of disease. You talked about the one type of seahorse you were working on, but does this affect lots of different seahorses? Does it impact pipefish and other fishes as well?
Yeah, absolutely. That's a great question. I'll let Jean-Philippe maybe answer the question because I know he has this topic in mind as well, and I know he's logged on. So go ahead.
Thanks, Claire. So yeah, I would say we study big bellied seahorses, but yeah, all seahorses are predisposed to mycobacteriosis. Hippocampus du Québec adds some back in time. Claire, you can correct me if I'm wrong, but yeah, other species also were affected by this disease, not only big bellied seahorses, but we use them because they are the second most popular species back in America. So that's why we use them for the study.
Are there any other questions?